Hey there, everybody. My name is Matthew Lee, and I am a second-year student at the University of Georgia, studying biochemistry and molecular biology with a minor in Spanish, as well as attempting to earn a master's in comparative biomedical sciences during my 4 times here at UGA. Today, I'm going to be talking about my project in which I deleted the KPR gene from various strains of Pyrrhococcus furiosus utilizing the CRISPR-Cas system. Now, the CRISPR-Cas system, it's a major technological breakthrough in the biological realm. You might have heard of it. It's extremely popular nowadays in many different applications, but it is a more recent discovery, and we're still learning a lot about how it can be used. As a brief summary, prokaryotes use it as an adaptive immune system in terms of adapting a short fragment of DNA from a virus into the host CRISPR locus near the leader, and eventually transcribing that and cleaving into multiple individual CRISPR RNAs. And these CRISPR RNAs eventually complex with Cas proteins, and this is where the adaptive immunity comes in. If the host is invaded by the same virus again, the CRISPR RNA will guide the Cas protein based on its genetic sequence, and the Cas protein will cleave that virus, thus rendering the invasion moot. So we wonder, can we use this CRISPR-Cas system that prokaryotes use for adaptive immunity, can we use it to delete genes and create new genetic markers for future research in our lab? So we wondered we chose the KPR gene, and we hoped that through a series of transformations with a specific mini array that targets the KPR gene, would be able to delete this. In the materials and methodology section, you can see a table with the strains of Pyrrhococcus furiosus that our lab generally uses. There's a wild type strain, we have an immunity null strain in which all CRISPR-Cas system is deleted, and then we have a few strains in which there's only a single CRISPR-Cas system. We have the 1A, 1B, and then a few variations of the 3B system. Now, how did we do this experiment? How did I go about this? So first, what we did was we transformed our various strains of Pyrrhococcus furiosus with plasmids that we grew up in E. coli. And these plasmids contain the PRF gene, which codes for the production of uracil, as well as either a randomized mini array that shouldn't target anything, and a designed CRISPR mini array designed specifically to target the KPR gene. So by transforming our strains of Pyrrhococcus with the plasmid, we allow the DNA to recombine into its chromosome, and what we hoped was that the strains that were transformed with the plasmid that contained the CRISPR mini array for the KPR gene would be expressed, and eventually those host cells would develop CRISPR RNA that'll complex with the Cas proteins and then actually target its own chromosomes that had the KPR gene using that mini array. And eventually the remaining chromosomes would replicate back to n equals 20 so that the cells would survive, only with the KPR gene deleted. So eventually the way that we tested to see whether the gene deletions were successful with liquid cultures of our cells after transformations and plating for selectivity, we extracted DNA from the liquid cultures, I amplified it by PCR, and then I screened it through gel electrophoresis. And you can see the results of that in the top right. So you can see the way that I loaded these wells were for each strain I loaded 2 replicates of DNA from cells that were transformed with the plasmid with the CRISPR spacer, and then cells that were transformed with the randomized spacer. And I labeled the different phenotypes. You can see that this middle type band is the wild type band. When the PRF gene was inserted through the pop-in, pop-out method, you have the upper band, and then with targeting and removal of that, where you have the simple KPR deletion, that is that bottom labeled band. So we can go through the results for each strain. We see that for the most part, at least one replicate for each strain shows that transformation with the CRISPR array showed more efficient removal of the wild type. You can see that based on the presence of the pop in with PRF as well as the pop out of the KPR gene. going across the board. Of course, you have varying levels of DNA isolation and amplification, which is something that, I'm a beginning researcher. That's something I want to work on. But we see that, especially, for example, in, the CSD, CMR, there's a much higher pop-in and not much wild-type phenotype showing, which is nice. Now, something to note is that transformation efficiency, growth was much less efficient with the CRISPR arrays. That's something that in the future we want to look at. Another thing to note is that with the CMR HD strains, we see that there is CRISPR-Cas cleavage, even though there's an inactivated DNase domain. Now, we have a few theories about why that is, and that's something that I want to look at in the future. But overall, we can see that the CRISPR-Cas system, in terms of preliminary results, can be used to delete genes from the chromosomes of Pilococcus furiosus. Now, moving forward, it's about making sure it's more efficient and that we have more definitive and clear results every single time. To wrap up, I want to thank Dr. Michael Turns, my principal investigator, for being willing to take on a first-year student and guiding me through such an exciting field. And of course, I want to thank my postdoctoral mentor, Dr. Ryan Ketchbull. He is super knowledgeable in this field, always willing to answer my questions and explain the mechanism behind everything that's going on. So, of course, a big thank you to him, and thank you for listening.